So good morning, everybody. We've got some great training presentations and pretty much nothing else to say, so we'll just go ahead and throw it over to move on.
What's the name?
I did. This is, oh.
No.
Sorry, yeah, let me see. Is that
Oh, yeah, we can. So if you click
this one?
Yes.
Yeah, perfect. Okay. Okay, I don't know what I'm doing. Okay, we'll just do this. All right, good morning, everyone. My name is Muhan, and I'm a MD PhD student, and I'm also part of the TL1 training group. And so today I'm going to talk to you about some of the things that I've been working on as part of my thesis and also as part of the TL1 project, looking at TGFB signaling and prostaglandin metabolism and fertilization of ovarian cancer. So this should be more basic science oriented. So if you have any questions, please feel free to stop me if I'm not explaining something clearly. So there's a general outline. I'm going to talk just briefly about what my general interests are, and then a little bit about my PhD thesis project, and then what I'm doing as part of the TL1. So I'm a fifth year MD PhD student, so starting my third year in the laboratory, doing as part of the graduate biomedical sciences. My thesis mentor is Dr. Michael Miller, who couldn't be with us this morning, but he's a professor in the Department of Cell and Integrative Developmental Biology. Developmental and Integrative Biology is a long name. So generally, my broad areas of research interests are women's health, specifically women's reproductive health, which in itself is still a pretty broad topic. And so I focused it down to fertilization as part of my thesis project, and then also looking at ovarian cancer, which I'm looking at as part of the TL1 opportunity. So I'll talk about both of those more. So for my main thesis project, I'm looking at the mechanisms of TGF-beta and regulating prostaglandin metabolism and fertilization. So before I get started, I just want to introduce the model system that we're using. So in our lab, we use these little worms called C. elegans, and they're about 1 millimeter in size. There's a couple advantages of using this model system. So they're hermaphrodites, which means they'll self-reproduce and you don't have to make them to maintain your colonies. They have a clear hypodermis, which means that we can see the internal structures, as you can see in this picture. So this is also great for me in looking at sperm migration because we can label the sperm of males and look at how they travel in the hermaphrodite gonad. So in one of the experiments that we do, we take males that have been labeled with a fluorescent dye, so their sperm are fluorescent. We make them with the hermaphrodite, so this male deposits the sperm into the vulva, and then we can see how it migrates through the uterus here. And I'll show you some of the pictures later. So another advantage of using C. elegans is that they have a very well characterized genome. So this allows us to easily manipulate their genetics to kind of look at molecular pathways. So one of the main questions we want to understand is how the sperm is attracted to the oocyte. So a couple of decades ago, people thought sperm migrated through this competitive race model, where basically large amounts of sperm gets deposited into the female reproductive tract, and then the most lucky and most fit sperm finds the oocyte and fertilizes the egg. However, with recent advances in technology, people have understood that sperm actually responds to a number of different processes. So people have seen that the sperm can respond to chemotaxis, where they swim either towards or away from chemical stimuli. And then they can also swim in response to rheotaxis, whereby they tend to swim against the flow of the fluid. And then the third mechanism is through thermotaxis, where they basically can sense minute changes in the temperature gradient, and they tend to swim towards places of higher temperatures, and that's kind of around the oocyte. But all of these experiments that people have done are mainly in vitro assays, so outside of the organism, mainly because it's really hard to kind of look at internal fertilization in most mammals. As you can imagine, it'd be very difficult to kind of trap the sperm in mammals. And this is where our C. elegans come into play because they're easily, you can easily visualize a sperm in an intact organism. And so the two overarching questions for my thesis is, what are the signaling molecules that are involved in guiding the sperm to the oocyte? And then what molecular pathways are regulating the metabolism of these signaling molecules? And so using these T ligands, we've identified a very specific class of prostaglandins that are important in guiding the sperm to the oocyte. So this F-series prostaglandins we found to be very critical in this process. And here's a schematic of prostaglandin metabolism and synthesis. So as most of you probably know, so arachidonic acid is broken down by these cyclooxygenase enzymes, and that's the drugs that aspirin and NSAIDs are targeting. And these enzymes then convert this arachidonic acid into an intermediate PGH species, and this then gets converted by different prostaglandin synthases into these various subspecies. And so, and each of them have different functions, which I think is blocking. But the one species that we're interested in that we found to be important in these worms is this PGF2 alpha species. But interestingly, the worms don't actually have these cyclooxygenase enzymes. So one of the questions we want to answer is what pathways are the worms using to make these PGFs, because they don't have the canonical synthesis pathway that we know. So one of the pathways where we've identified is the ZAF7 TGF-beta pathway. I'll just use the pointer because the mouse is.
Oh, I don't think the pointer shows up.
Oh, I have a TV. Okay. Yeah. Well, it doesn't block too much. Okay. So so we found that this ZAF7 pathway, which is a homolog of the human TGF-beta pathway, can modulate sperm guidance. So here's a schematic of what we know so far about this pathway, and here are some of the experiments that we've done called the sperm guidance assay, where we look at how sperm migrates in the uterus. So we see that in wild-type conditions, when nothing has changed in normal worms, the majority, 91% of the sperm, targets this sperm holding site called the spermatheca right here. However, when we knock out certain genes like this DAF7 ligand, we see that the sperm has a significant defect in the ability to travel to the spermatheca. And then if we knock out the downstream receptor, DAF1, we see a similar defect. And similarly with the downstream DAF14, which is an RSMAD. However, when we knock out, when we basically eliminate the DAF1 receptor in a downstream antagonist of the pathway, we can get rescue of this phenotype. So what we concluded from this is that at least sperm guidance is modulated by a transcriptionally regulated pathway. So then we wanted to understand, now does this pathway also modulate prostaglandin synthesis and metabolism, because we see that this prostaglandin is important in sperm guidance. So again, so we did mass spec to look at these PGFs in the species. And again, we look at different mutants. So then the blue, you can see that is wild type, and we have a nice peak, PGF2 alpha peak right here. And then when we knock out the DAF1 receptor, we get a significant decrease in the amount of prostaglandins that we can detect. But then when we knock out, again, the double mutant, the DAF1 and the DAF3 together, we get a rescue of this phenotype. So what we're seeing now is that this pathway actually also regulates prostaglandin metabolism. So what we can conclude so far is that these F-series prostaglandins are important for sperm guidance, and that this TGF-beta pathway can transcriptionally regulate these PGFs in a COX-independent manner. And that's a very novel pathway, because what we know about prostaglandins and arachidonic acid metabolism is through this COX pathway, and now we're looking at a different pathway that's independent of this enzyme. And so for future directions for my thesis work, I want to characterize where this pathway is acting, so what tissues are involved, and then also look at the downstream targets of the pathway that may be important in modulating prostaglandin metabolism. Okay, so switching gears, so as part of the TL1 project, I want to look at this, TGF-beta pathway and PGF in ovarian cancer. So it's pretty well established that the TGF-beta pathway is involved in tumorigenesis. So in normal epithelial tissue, as you can see here, these cells respond pretty well to TGF-beta, but then as they progress to become more tumorigenic, they lose this responsiveness and they begin to synthesize more TGF-beta as a consequence to kind of trying to simulate the same mechanism as in normal tissue. However, let's see. Yeah, and then also, so prostaglandins have been seen to be involved in tumorigenesis. So you can see here people have looked at PGE as a subspecies to induce tumorigenesis and how it's involved in stimulating some of the processes that are important for tumor to grow, such as angiogenesis, and immune response, invasion, and such. So while we know that the TGF-beta and prostaglandins are important in tumorigenesis, we really don't know if there's any crosstalk between the two pathways to affect tumor growth. And so that's kind of what I want to look at. And excuse me, to answer this question, some of the questions that we want to answer as part of this TL1 is how do PGFs modulate tumor behavior, and then does this TGF-beta pathway regulate PGF metabolism in tumor cells? And so to answer these questions, we decided to use ovarian cancer as a model. So high-grade papillary serous ovarian cancer is the #1 killer of all gynecological cancers. 70% of women die within 5 years, and almost all of them die within 10 years of diagnosis. So this is a very deadly cancer. And mainly it's because most of the people who come to find out that they have cancer are already in the late stages, where it's very hard to treat. So of all the ovarian cancers, 90% of them are epithelial tumors. And beyond what people use now, such as the platinums and Taxols, there's very limited therapies. And there's actually a lot of clinical trials ongoing to find novel therapies that they can either add or switch out from the regular platinum and Taxol treatment regimen. And so we've also found that PGFs are in human follicular fluid. So this suggests to us some tissues in the female reproductive tract is secreting these PGFs, and we have evidence to think that it might be the ovarian cells. So for a research strategy, what we plan to do is take a series of ovarian cancer cell lines and screen them for PGF synthesis to see if this pathway is even active, potentially active in these cells. And then we plan to use drugs and genetic approaches to kind of delineate the effects of this PGF on ovarian cancer behavior. And then to also use drugs and genetic approaches and manipulations to kind of look at the effects of the TGF-beta pathway on this prostaglandin metabolism. And finally, what we hope to achieve as part of this project is to actually analyze human ovarian tissue to look at healthy healthy ovaries versus cancerous ones, and to assess the association between the PGF and the TGF-beta ovarian cancer. And so with that, I'd like to thank, so my lab here, as shown here, is Dr. Miller's lab, and then the Temple Core has been very good. If you guys ever need to do mass spectrometry to look at proteins or lipids, they are very good at that. And then the cell line we're doing is part of a collaboration with Dr. Rebecca Aaron's lab. She's a gynec professor. And then thanks to my funding sources. So if you have any questions or recommendations, suggestions on directions I could follow, I really appreciate that. Thank you.
I actually have a question. So like the first part you were talking about, you guys, I don't know if you discovered it, I don't remember what you said, but the new pathway about prostaglandin formation, like that's, and I'm a pharmacist, so I see like drug development as being very important there. Like, have you guys dabbled in that? I know it's very random and all.
Yeah, so actually we're still at the stage where we're trying to figure out what protein is responsible for it. We just know that they don't have these COX enzymes or the regular canonical pathway enzymes, but we haven't. So the pathway that I'm showing you isn't really enzyme targetable, just because TGF-beta is so important in so many processes in the body. But what we hope to find is that by finding downstream targets that are more specific to this process, that we can potentially find a target in the future.
Is that pathway also in mammals, or is that strictly a C. elegans?
So we think it is. So we have generated COX-1 and COX-2, so there's two COX isoforms in the body. We've generated knockouts of both, double knockouts of COX-1 and COX-2, and we still find these PGFs. So we don't see any of the PGEs, PGDs, or TXA isoforms, but we see that, even though they're really unhealthy, but we do still see PGF2 alpha. So it's telling us that, you know, independent of the COX enzymes, we still have this pathway to make at least this one very specific species of prostaglandins.
Other questions?
Thank you.
Thank you.
All right, so my name is Katella. I'm a 5th year in UAB's Medical Clinical Psychology program. So I'm a TL1 scholar, and as a part of that, I'm working on my dissertation in the field of pediatric psychology. So clinically, I have worked extensively with the Division of Hematology and Oncology, and they have a very strong psychosocial program. It includes things like child life specialty, pastoral care, social work, as well as psychology and neuropsychology. And I've noticed that these resources aren't consistently offered to other departments throughout the hospital. So I've done some research work with neurosurgery with one of my mentors, Dr. Rock is here, and I noticed that a way to combine my interest in oncology with my interest in developing research, looking at psychosocial care delivery within hospitalization, is to look at these patients who had benign brain and central nervous system tumors. So I am looking at low-grade central nervous system tumors, which is defined as those that are treated with surgery only. So no chemotherapy or radiation. And for the most part, they're pretty much considered to be pretty harmless, and there's not a ton of follow-up done on these children. And when you look at the literature, for the most part, there's not any, severe devastating side effects like you would see in their counterparts that receive chemotherapy and radiation. So they're mostly assumed to be harmless with limited side effects. However, there's some preliminary research out there to suggest that they actually do have some effects that could affect quality of life and mood outcomes. So, for example, one qualitative study found that 56% of their sample actually had problems in anxiety, depression, behavior, or academics. So this is an area that I think is really important to look at and to really figure out if this is a population that we should be delivering more services to. So my dissertation has 3 aims. The first is to characterize quality of life and mood outcomes in this group. So this is the first study to look at this from both the caregiver and child perspective. We're looking at those who have had surgery within the past 8 years and surveying caregivers with a child of any age and then children between the ages of 8 and 18. So looking at measures of quality of life, brain tumor-related effects, so cognitive problems, pain and hurt, emotion, imbalance problems. And we're hypothesizing that children after resection of these tumors will demonstrate significant quality of life and psychosocial difficulties. So not to the extent of their counterparts who may have received further therapy, but definitely more than the general population and their healthy peers. The second goal is to determine if there are demographic or medical characteristics that predict problems with these outcomes. So complicated treatment course, brain tumor location, things like that. And of course, we're hypothesizing that patients with a more complex medical course will encounter more difficulty, and that broadly the diagnosis will affect the family beyond resection more than it's considered now. Finally, I want to look at the psychosocial services offered to patients and caregivers during hospitalization and to determine whether quality of life or psychosocial outcomes are predicted by the services that they've received during hospitalization. My guess would be that children and families who receive more services will demonstrate better outcomes, but also that those who are seen by services that have strong psychosocial support, such as oncology or rehabilitation, will have seen more services and perhaps have better outcomes from the perspective of how the diagnosis affects their life. So I am currently collecting data for this project. We're looking at the caregiver perspective of the patient's quality of life, mood, behavior, and also the services received during hospitalization. So we just came up with a measure that asks, did you see these different services? Were they helpful? And kind of how did you utilize them? So, and from the patient perspective, we're also looking at their anxiety, depression, and anger. Preliminary results suggest that, so I looked at the data I've collected so far, which is 30, and I submitted it to the Society of Pediatric Psychology annual conference. So when we looked at another study that was published using the PEDS-QL and looking at a healthy population kind of during their well visits and a chronically ill population, our data really lines up with those who have chronic illnesses and are significantly worse than their healthy peers. So this could, I mean, looking at my sample, it definitely could be those that have comorbid conditions or have encountered more difficulty are the ones more likely to seek out this kind of research and participate in it. But either way, I feel like this really is pointing in the right direction of that this group needs to be studied a little bit more. Questions?
How are you recruiting people for the study?
So I looked at the registry of the surgeries that have been completed in the past 8 years, and they're being recruited either, I sent out an invitation to all of them. They can complete either during their routine clinic visits, which for some kids is annual, and the other option is to do it electronically.
So one comment, I think you really could have made your typeface bigger.
Okay.
So I think that, you did a really nice job, like your titles were nice and clear, but you had a lot of white space. You could have made the rest of it a little bit bigger.
Sure, thank you.
I had a quick question. It's a great study. I'll be interested to see once you get more of a sample size too. And what are some of the what do they do with in that clinic for the caregivers? Typically for psychosocial, is it any support or is it more based with the children? Or
typically so some of them are followed by oncology, in which case they more routinely will have these services available in the clinic. For neurosurgery, if there is a significant problem, they would refer them to the oncology psychosocial program, but there's nothing routinely offered to these families at this time.
Yeah. It's a good study because the ongoing, they're little kids, obviously lifelong issues.
Mm-hmm.
So,
I guess more of a medical terminology question. So how are you defining benign tumors? Is it just
that they didn't?
I'm sorry.
Sorry, just that they didn't receive further chemotherapy or radiation, surgery only.
Okay. I guess a comment would be, I don't know, I guess Dr. Rockins exists, but I don't know if I might steer away from that term. It's a little loaded to me. Benign.
The benign tumor
Because,
term?
I don't know. It's, just simply that you don't need radiation or chemo doesn't mean that it's necessarily benign.
That's a valid point, yeah.
I don't know. And it might,
That's what our hypothesis here, that this is not necessarily benign.
exactly. I mean, that could even, maybe it's something for your discussion when you get to that point, is that maybe we shouldn't be calling these tumors benign.
Yeah, I agree.
Because we wouldn't call a high-grade brain tumor benign, although technically speaking, you typically are benign if you don't have metastatic potential.
No, so a high-grade brain tumor would be considered a malignant tumor. It's only...
Well, no, I'm saying, but if you were just, I'm saying, I know we don't consider it benign, but strictly speaking, it still doesn't have metastatic potential.
Brain tumors are a little different in that they don't metastasize outside of the CNS.
Right, exactly, yeah.
But
So it's a little bit of a gray zone.
The point is that the way that she's structured this, that the kids who end up or who are getting chemotherapy and radiation are routinely offered psychosocial support.
Mm-hmm.
The kids who get surgery alone aren't. That's how we've defined the sample population here. That they're
No, I
in
think
the surgery alone.
it's a good design, it's just the term is
Yeah.
a little
Right.
That's a
misleading.
great point. Yep.
I actually have vacillated between, for the most part, I've referred to them as low grade.
Yeah, that sounds more,
But
I don't know.
I've just gone back and forth because to me, when I say that, some low grade tumors might receive chemotherapy or radiation. So I don't know. It's a good point though. I agree with you. Yeah.
I
think actually you did a nice job of sort of saying, generally these are considered benign, but, here are the things that we think could be an issue.
Right.
So I thought that, you did a nice job with that sort of setup in terms of, getting people to buy into, this is a very important issue, people who don't necessarily know anything about it. So I thought that was very good.
Thanks.
I think you raise a, oh, sorry. A real quick question. I think you raised a good point too. I'm kind of curious too, when working with different patients, families, what they perceive and understand about like what diagnosis, what that means, benign, malignant, and just talking with patients,
Yeah, and just
the family members.
and that's part of what inspired this project is just talking to families. Even when there isn't further treatment, there's still this tremendous anxiety about it, and they still worry, will this come back? Even if, medically there's really no reason to suspect that, it's still a lingering anxiety within parents. Just clinically, I've observed that. So, yeah, I'll be interested to see also.
So, sorry, did somebody else have a question? So it's kind of a methodological question, so I don't normally focus on adolescent and child-based research, but essentially, so I was a little curious how the composite quality of life score was. So is it kind of combining parental and child perspectives on?
No, they'll be analyzed separately, and I'll be interested to see if they correlate. If they don't, that'll kind of change how I look at things. But
yeah, I think my one thought was that maybe potentially if the sample size permits, it may be worthwhile to look at maybe the older subset of children or adolescents and then comparing the younger because their perspectives may differ on the quality of life measures.
Yeah, I agree. Age is definitely a component that I want to look at.
Thanks. Just curious if you think the impact on quality of life is more specific to where they're having surgery, such as having brain surgery versus just having a major medical event in childhood, and if you consider using another type of a control group that would be an orthopedic injury or some other type of injury outside of central nervous system.
No, that would be really interesting. I was thinking of it just in terms of psychosocial care delivery, comparing it specifically, thinking about brain tumors who do go on to be treated in oncology and those who don't. But that would be also another interesting direction to go into, is looking at it as kind of just a major medical event. But I guess I'm just, in my mind, or the way I conceptualize it is as something that could be more chronic than that. It could foreseeably return, and they'd have to. Does that make sense?
Yeah.
Okay.
So when you're thinking about the groups, is there a difference in the follow-up as far as the ones who don't have a tumor that goes on to more treatment? Do they still continue to come back to the clinic?
No. That's a simple answer. They'll come back within, they'll come back soon after surgery, and then 3 months after that, and 6 months after that, and it will kind of taper off.
So the idea to, provide this support at the very beginning?
At the very beginning, yes. And then if there are identified issues, that could continue more frequently, or we could make community referrals. That would make more sense for them to continue, when they go home.
The kids who are just seen in neurosurgery, we see them frequently initially after surgery, but then on an annual basis. And it's you get a scan and you see me for 5 minutes. Oh, your tumor's not back there. Are you doing okay? Okay, great. See you later. We don't offer anything substantial in terms of psychosocial support. The question is, should we?
Right, and it's not, it's not that it's, if there is a major issue, it'll be identified, but it's not routinely assessed. They're not saying, how stressed are you? How is this impacting your life? In general, I'm not really sure if school difficulties are assessed.
It's literally a 5-minute visit.
Yeah,
It's the routine right now. So this study could very well push us to a much more routine assessment of psychosocial needs.
Mm-hmm.
Thank you.
Thanks so much.
Mine's still on this piece.
Okay, you can go.
Storage podcasts that I subscribe to download if they don't know what's trying to believe in. But I have space on the phone. It's hard to play.
All right, so my name is Matthew New. I'm a fourth-year MD PhD student, and I actually live in Huntsville and work up at Hudson Alpha in Greg Cooper's lab. So I'm normally one of the faceless names that you see sometimes asking questions on the screen up here. So my PhD thesis project is actually in basic genomic sciences, doing an intensive computational analysis on a very large autism cohort. And I've been in the TL1 for a year now, and when I joined, I kind of wanted to do something a little bit different from the basic science to look at some outcomes research, effectiveness, and to kind of get a perspective on the, from a patient's perspective, on the more clinical end of the genomics research and diagnosis pipeline. So to that extent, have been working with Dr. Sarah Knight and some of her projects that she's been working with, actually partially in collaboration with my thesis lab, has a tail end of getting information from patients regarding genome sequencing and some of the results that they receive. So in advance of some of those projects, we set out to do a literature review in a organized fashion, initially with a scoping review, which I'll discuss some of the very brief results from, and then moving on to a systematic review of the literature as well. So it's a little bit different than the other projects that have been presented today that are more hypothesis-driven. So bear with me for a minute, and it'll be fun. So some motivation. So imagine just for a moment that you are a clinical geneticist, and there's a 2-year-old girl that's referred to your genetics clinic right here, very adorable, due to developmental delay, facial dysmorphism, and some sort of autism-like behaviors. So the clinicians in the room might be thinking, like, oh, what could this possibly be? But think about this. So both of the parents are healthy. So they do you have a previous single gene test that's negative for something that's somewhat common cause of these types of features in girls for Rett syndrome and the MECP2 gene. So what's your next step? You don't have any positive results? What do you do next in the diagnostic pathway? Well, you could order a microarray. This has probably been done before, but you do it anyways, to look for any sort of chromosomal abnormalities, any sort of microdeletions, and it's totally negative. So you could individually sequence more genes that you think could be related. But to avoid this diagnostic odyssey where you could endlessly sequence genes, you encourage the entire family to get whole exome sequencing. So part of the strength of getting the whole family to do this and to participate is that you can look at inheritance status. And keep this in mind for a moment. So just to get everybody on the same playing field, what is whole exome sequencing? Whole exome sequencing is a next generation sequencing technique that captures all of the protein coding regions of the genome. So it's only 1% of the genome, but most would argue that is the most important part of the genome. So in your 2-year-old girl, you find, after this analysis, that there is a non-inherited frameshift mutation in NIPBL, which is a gene associated with Cornelia-de-Lange syndrome. It could explain her phenotype. And the parents are relieved. They're ecstatic to finally have an answer for their child. After extensive testing, a lot of questions, they finally have an answer. But unfortunately, remember you sequenced the family, and you did an unbiased test. So you got a little bit more information than you bargained for. You find a couple extra things, some incidental findings, which are findings unrelated to the primary reason why you did the sequencing. You found that the father has a BRCA1 mutation, which is a gene highly correlated with development of breast cancer. You find the mother has an APP1 mutation associated with an early onset of Alzheimer's. So, what do you do? What do you think? Do you return these results to the parents? Do you really think a male is going to get breast cancer? It happens. Almost all of the cases of breast cancer in males are due to BRCA1 mutations with a strong family history. What about APP1? There's nothing we can really do to prevent Alzheimer's in somebody. would it cause undue stress to them to return these results? So fortunately, we actually have some guidelines from the American College of Medical Genetics, and related to the project, but also to an extent unrelated to the project, the BRCA1 result would actually be, theoretically, you would have an obligation to return it, but APP1, not necessarily. So this motivates the question, well, the obvious solution to this is, well, you consent the patients beforehand and you ask them what they want, okay? And I can tell you from projects that we've had, sometimes when you consent the patients for something that, something like this, they say one thing, and then when they get results back, they have a lot of regret about it, about the selections that they made during the consent process. But in any case, so this is kind of the motivation for better understanding what are the attitudes and preferences towards patients to this type of test. So the project aim, as I kind of previously discussed, is to simply perform this literature review to understand what's currently known in this area. So first, I performed a scoping review. And this is a less formal type of literature review to essentially just give an overview of the type, the extent, and the quality of available research in a given area. So briefly, the methods were to simply look on PubMed, only one database for this, using the terms WES, which is common abbreviation for whole exome sequencing, whole genome sequencing, attitudes and preferences, and to look at recent studies only. So with Dr. Knight, we were able to whittle this down to 13 papers, which is a very small number because we used very precise criteria for this, were identified meeting these criteria. Of these, very few of them actually looked at, or only less than half looked at patients themselves. Some did a community sampling, and others actually looked at health professionals to thought what they should be returned to patients themselves. And no publication included in our study targeted any sort of medically underrepresented population either. So what is the sampling technique for the existing literature? Almost all of them, as you can see from this overwhelming pie chart, were convenience sampling. And only one that we looked at was some sort of attempt at randomized representation of the US population. So publication by journal type, almost all of them are specifically published in genetics or genetic counseling journals, so they might not necessarily be reaching a wide audience. One publication we found in an oncology journal, and one was in kind of a general medical or even a psychology journal. And then looking at just the sample size by publication, there's a huge array of the number of participants that they were surveying in these studies to ascertain their attitudes and preferences. And it ranged widely from, as you can see, 16 to almost 7,000 individuals in one study. So when they looked at what is the primary metric of interest, what is our study really trying to look at, they saw that the majority were looking at incidental findings, which are these findings that I kind of give an example of that are unrelated to the primary reason for doing the sequencing. Some also looked at just the general, how interested are you in receiving whole genome sequencing, but far fewer than those looking at incidental findings. So some of the conclusions of this initial scoping review is that the existing studies are limited in their representation of the US public, the journal audience, the sample sizes, and then the patient representation as well. So most of these studies focused on the return of incidental findings. None of them looked at medically underserved populations, and the interest of individuals wanting to receive incidental findings that are, again, like the example that we discussed, is high, ranging from roughly 85 to 99% in publications included in our study. So moving on to that, we have decided to extend this into doing a systematic review. And well, what's the difference? Is that this is a more formal literature review that more critically analyzes a selection of papers using a predetermined structured method. You frequently actually have to submit some of your methodologies to databases and get almost like an approval before you do the study. The idea is to state what you're going to do up front and then follow through with it based upon a predetermined information document. So some of the current progress on this is still an ongoing project, and a big thanks as well to Jamie Richards, who's one of our collaborators at Hudson Alpha, who's also helped out a lot at this point. So we've generated this Prospero protocol, and this is a register of systematic reviews that you would submit your plan for the systematic review, as well as to refine search criteria that you would use to input into publication databases such as PubMed. And this is a much larger task than you would possibly conceive it to be based upon a simple PubMed search that we do daily, just looking for information. This is more of an iterative process to have the greatest sensitivity and specificity for inclusion of publications into your review. And so just to, I think I have an example here. So this is one of the searches that we ended up settling down on. And I don't want you to actually read it because it's not that important, but it's essentially chaining together a whole bunch of terms and then trying to use some exclusion terms as well. And then, so this search term yielded roughly 285 publications. And then we can refine it continually to get for 154 publications here for this monstrous chunk of text. So the question is, you might have is, well, how are you supposed to determine sensitivity and specificity to something like this for relationship towards what you're actually trying to assess? And the answer is, it's not like there's a gold standard for this. At some point, we're actually going to just go and read the abstracts to see if it's really the type of search that we want. So beyond this, some of the next steps for the actual systematic review is to generate a final list of included publications and then to actually go through, Jamie and I will actually summarize some of the publications while extracting relevant data. So that's actually all that I have right at this point. So it's that second part of the project is still ongoing as well. So some special thanks to Dr. Knight and Jamie Richards, as well as UAB, MSTP, and then the TL1. So I'm happy if there are any other questions that somebody has. What you got?
I really appreciate the introduction. Even as a clinician, it was extraordinarily helpful to sort of put my figure on a patient, this and that, and so forth. So
sure.
that was very helpful.
Great. Yeah.
Which type of databases are you thinking about searching? That's question number one.
Mm-hmm. Okay, so obviously the primary database is PubMed. We're intending to use some of the other databases, JSTOR, Google Scholar. Yes, that's one of them as well. But as far as we understand, and we checked a couple of them just doing some sort of preliminary searches, as you can imagine, PubMed is going to represent 99.9% of any of the papers that we want to include.
Do you have a librarian helping you with
Yes,
that?
we do. Mm-hmm.
So UAB has now access to embeddings. We used to not have it before. Now we do for free. I would be cautious about searching Google Scholar because it just searches for everything. The quality of the data may not be that relevant.
Right. One of the other recommendations we were given was also to search for non-English publications. And then if we find non-English, that actually strengthens the protocol that you write, we're told, at the onset of the project. But that has its own things with it as well.
And the other one is the Scopus.
Yes, that's correct. We also are searching Scopus. All right, cool, thank you. Oh, sorry, yep.
No, go ahead.
Well, I just have a waxing philosophical question.
Please, this is my favorite.
Which is, I keep thinking about your pie chart and how sort of it was, overwhelmingly not randomized. And is that a bad thing, right? If sort of whole genome sequencing is really meant to be like a step to personalized medicine. Do we want to randomize that?
Right. Well,
That's
so
just my philosophical question.
At this point where whole genome sequencing is fairly expensive as a test, and it's rapidly decreasing in cost. We can do Hudson Alpha. Don't quote me on this, but it's roughly 2,000 or so for research genomes to be pushed through, a little bit less perhaps. And exomes are even cheaper than that. Clinically, it's a little bit more expensive. On the research end, that's where we're at. And towards the push for personalized medicine, we are starting to do next generation sequencing, whole exome, whole genome sequencing on healthy individuals. But to this point, most of the sequencing that we've done to this date, in fact, have been on individuals who have some sort of health complication that we're trying to assess. So I think that has a lot of motivation for the patient selection as well. There are people that are in a hospital that are part of a research project that are already getting sequencing for something else, something health related, which is, again, part of the explanation why we don't have much of a randomized population sample. So to answer your question, yes and no, but it becomes more important the more interested we become in sequencing healthy individuals that would just be in the population.
Do these papers, in your scoping review, talk about how results are returned and, to what
Yes.
extent people are getting interpretation versus just results?
Absolutely. So one of the questions that many of the papers address is they ask a survey question like, who would you like your results returned to you by? A genetic counselor, a geneticist, your primary care physician, or other, right? And I think most of them would, it's kind of an admixture, and I don't remember any of the direct numbers off the top of my head, but most of them would want to hear back from either the genetic counselor or actually their primary care physician, which does motivate another question, is that most primary care physicians are not very well versed in whole genome sequencing and the pitfalls of doing whole genome sequencing, the results that can arise from it, as far as, you can get into a quagmire of not fully penetrant information, of false positive results, et cetera, et cetera. So it does take a bit of more sensitive understanding to this. And I think it actually begs the question of, for primary care physicians that could be returning these type of results, to have an education and understanding of the details of it better.
I have a couple more questions and a suggestion. So one is, my understanding is you want to explore attitudes and preferences for this type of testing, correct? Did I get that from the beginning?
Absolutely, yeah.
Right. So do you have a sense as to which variables you want to abstract from each one of however many papers you obtain?
Right, so some of the variables can include stuff like just general interest, how many people are interested in even having the testing, who wants to be the communicating individual for the results, and then what type of results are they interested in. A lot of papers, for me personally, some of the more interesting items would be, would you like results returned to you that are not medically actionable, that are medically actionable, that are medically actionable in your child, or not medically actionable in your child? To me, those are some of the more interesting numbers, but I think it really depends on what we end up getting as far as the papers.
So what I'm hearing you saying is you don't really have a data collection form because you don't know what's going to bubble up from the studies.
To an extent.
Okay. So think about that carefully, because you're going to spend a lot of time putting papers, searching this and that, reviewing abstracts, and then you're going to receive 20 to 100 papers. And then the difficulty is going to be which information you're going to extract from which papers, especially when this is sort of breakthrough in terms of new information.
Right.
In that setting, the systematic reviews are a little bit harder to do because you don't have measures that you can aggregate. You don't have like different ratios, sensitivities, or anything. So these may turn out to be a narrative review and not a systematic review. Or you can leverage, and this is the suggestion, you can leverage the framing for the scoping review to fit this need. We just are midway through scoping review for another purpose, and it was extraordinarily difficult to just do exactly what you were talking about,
right,
to the point where we don't know what we're doing in that sense, just because it's so difficult.
Sure.
So my suggestion would be leverage your quick review, and if you can do a scoping review based on that, instead of embarking into sort of a really long journey if you don't have a structure to guide your data collection. That's all.
All right. I noted that one of the challenges of doing the scoping review and trying to collect some of the information out of them was that each one of the papers that we included asks just a mildly different form of each question. So it's not like a clinical trial where they were very clear and exactly if drug A or drug B was more effective. They'd ask just a slight variant of the question each time. So trying to extract some of that that almost metadata from it is certainly challenging, but something that we're up for the challenge for. Cool. All right, great, thank you.
Okay, hello everybody. My name is Sarah Cable, and I'm going to be doing kind of a wrap-up presentation on what I've done this past year with as a TL1 trainee. So kind of the title of my research project is Examination of Parental Communication Patterns and Satisfaction with Care Following Youth Concussion. So an overview of what I'm going to talk about today, give you a little bit of background information, my overall aims for my research, the study design, a few kind of results of what we have so far, some conclusions, limitations, and then kind of just some lessons learned through this TL1 trainee process as well as doing this research project. So some background here. So youth concussions, about 1.1 to 1.9 million sports and recreation-related concussions occur in children a year. And a concussion is a type of traumatic brain injury that occurs from a direct or indirect force to the head. Sports are the second leading cause in ages 15 to 24 after motor vehicle accidents, which are the #1 cause. So you hear a lot about it being in sports, but it's important to remember that it's not just sports that concussions are occurring in. So some sports are the highest rates of concussion are, of course, football, girls soccer, boys ice hockey, and boys lacrosse. Rates of emergency department visits have significantly increased over the past 5 to 10 years, and that's just kind of related to greater awareness of concussions. And then also kind of put things in context, orthopedic injuries, other sorts of sports-related injuries make up a much greater portion than concussion injuries. So only 7% of sports and recreation injuries are actually concussions. So concussion treatment, it's kind of just, there's a lot of different guidelines, consistent statements of how to do this, but it basically all boils down to physical and cognitive rest, followed by a gradual return to baseline activity. And within kind of the physical component, so returning to play, returning to activity, there's been a lot more research on that. The guidelines are a little bit stricter and a lot more people agree with them, kind of well known in the medical community. But then when it comes to cognitive rest or getting back to school, getting back to thinking, there's a lot of variability. Not too many empirical studies have been done on this. So it's kind of an area that we need more research on. And then a majority of children will recover from a concussion within 4 weeks. There used to be the statistics of like 10 to 14 days, but we're moving away from that with children and adolescents because that statistic's more so for adults. So now kind of for children, we expected them to recover within 4 weeks, but about 10 to 15% may take longer. So kind of we're really interested in this return to learn process with this project, so that cognitive rest, getting back to thinking, back to school. And like I already mentioned, there's not a lot of data on this, the best way to do this. Some studies show that too much cognitive rest can be bad, have poor outcomes, take longer to recover. And then some other studies show, well, there's children going back too soon, and that negatively impacts the recovery. And of course, cognitive performance is often impaired during recovery from a concussion. Schools are not the best place when you have headaches and are sensitive to light and noise. The hallways are crowded. Those fluorescent lights are really bad. You might have a headache, which interferes with your concentration. And then usually following a concussion, fatigue's a major issue. So if you're really tired, you're not going to be able to learn as well. So the systems of care I've been looking at with this project are here. So we have the family, which of course is the child and the parents and siblings too, and then the medical system. And that's not just physicians. Sometimes kids need to be seen by optometry or by physical therapy, different groups that you might not always realize are involved in concussion care. And then the school is a major component, especially for return to learn. And the individuals within that kind of that system are your teachers, of course, school nurses, principals, guidance counselors, all those individuals. And then the sports and recreation, which are your coaches and athletic trainers. So a few studies have looked at communication across systems of care, but no studies have really taken the parent's point of view. So there's been a few papers on like asking teachers who most frequently contacted them about concussions, and they said school nurses and parents were those. Nurses reported being contacted most by parents. Another study said that school athletic trainers don't really communicate with the other school personnel that are more involved in the academic side of things. And then some school nurses reported barriers to concussion management as the lack of communication with the medical professionals that are kind of overall caring for the child. So school nurses don't know what the physician's saying and what they need to be doing in the school setting. And like I already said, nothing really done on the parent's point of view, which is what we're doing here. So the study aim one is just to kind of characterize these parent communication patterns between the systems of care. So looking at the frequency, how often are they talking to people? The method, phone calls, text, email, in person, how are they doing it? Who exactly are they contacting? Are they going to the principal first? Are they going right to the teachers? Is there a point person at the school? Those sorts of things. And then are they satisfied with communication? Do they feel like the person they were talking to understood what they were saying, understands the child's limitations, and what might need to be done acutely in this process? And then how often are they even asking their kid, like how their kid's feeling? And then aim two is to examine the relationships between parent communication with systems of care, and then just any youth and parent associated demographic factors, medical factors, and psychosocial functioning. So some child factors that might be related to frequency of communication are how long it takes for the child to get back to school, so days to return to learn, how severe symptoms they're reporting at first, age, race, any history of psychological or learning disorders, number of previous concussions, and then the emotional well-being of the child, and then parent factors, age, education, income, employment status, and marital status. And the results I'm going to present to y'all today are more aim one. Aim two I'm going to get to later. So study design, just to get an idea of what we're doing here. So screening for potential participants, we're recruiting them within 14 days of injury because we want to kind of get this kind of acute idea. If we're asking parents later on, well, who did you go to first? They might not remember accurately, especially not remember accurately how many times they may have communicated with someone. So as part of our kind of initial intake, we're having the youth do the youth inventory, anxiety and depression scales. Also, the SCAT-3 is kind of just a symptom inventory of concussion symptoms and severity. And then we're getting from the parent how they perceive their child's symptoms, just basic demographic information from a family information sheet, and asking them communication from the time of injury until they're enrolling in our study, which is no more than 14 days. Then after that, we are tracking this communication across time. So we're calling the parents and the child once a week for at least 4 weeks and asking them, again, kind of the same things, symptoms, asking the youth, how often have you been talking to your parent about your concussion and related symptoms? And then for the parents, asking them, how do you perceive your child's symptoms? Child's progress in school, so have they fully gone back to school? What sort of accommodations are in place as of now? And then how often have you communicated with these different systems of care? So I kind of mentioned earlier that not everyone's going to recover within 4 weeks. So if they have not recovered within 4 weeks, they're considered prolonged. And we do 4 more calls, and we actually do those every 2 weeks to kind of follow them more through this process. If they've recovered in 4 weeks, though, study's done, and we have that. And this is that symptom inventory I was talking about, so all your different concussion symptoms, headache, pressure in the head, feeling like in a fog, difficulty concentrating, difficulty remembering, and they can rate those 0 to 6. And this, I guess, hopefully you can see this okay. This is kind of an example of what our communication form looks like. Now, this is a lot, so we don't actually hand this to the parent. We do this kind of interviewing them, and I'm the one filling it in because this is just a lot to look at. And here's kind of the principal, the school nurse, school psychologist, guidance counselor, teacher, school office aide or secretary. We didn't originally have this one, but then a lot of parents were like, oh yeah, I just gave the forms to the front desk lady. And we're like, oh, okay. So we kind of made it its own thing in here. And we're asking the number of times you attempted to contact or contacted, the method, satisfaction with that communication, and then what did you talk about. So the current status, we have 37 participants that have finished. There's another 9 currently enrolled, and actually have a participant this morning after this talk. So there's a number of, lots going on right now. And then the percentage of participants that completed every weekly call was about 80%. So it was, we missed some weeks with some parents. We just weren't able to get a hold of them. And so here's the child demographics with this sample of 37 we have so far. Average age is about 14 and a half. Symptom score when they enrolled in the study. So there's actually 2 different scats. The child scat uses different symptoms, a little bit of a different scale. It's 0 to 3 instead of 0 to 6. So I separated them. So this is, they're endorsing 8.5 symptoms. On the child scat, they're endorsing about 11 symptoms. And then the severity, this is for the scat 3, that's out of 132. So 19 is what they're reporting as far as severity goes. So that's adding up that 0 to 6 for all the symptoms. And then on the child scat, about 21 for that initial severity. About 62% male, the rest female, primarily Caucasian, primarily in middle school and high school. And then most are at a public school, but some are at private schools. And then the majority of concussions did occur during a sporting event, game or practice, but some others occurred with just different physical activity. Again, majority of participants are athletes. Psychological history, about 24% have ADHD, some others have anxiety, depression history, and then the average days to return to learn, 19.3 days, and then 7 out of that 37 fell into that prolonged category that we call an additional 4 times. And then parent demographics, so average age, about 44. Of course, this shouldn't surprise anybody, mostly moms that were being the ones doing the communication, that were willing to talk to us over the phone and stuff. Same racial background, majority married, very well educated parent population, so a good number with graduate degrees, postgraduate training, and a lot of these parents who are working full-time as well, or at least working part-time. So here we have communication over time, and I just have initial through week 4 because there's only 7 that we did the weeks 5 through 8 calls with. So this line here is your medical. How often, percentage of parents that communicated with any medical professional when their child was reporting symptoms. So the sample changes because if the child is reporting 0 symptoms, that's kind of a way to say, well, they've recovered more or less. So you wouldn't expect them to be communicating as much if they recovered, especially with like medical professionals. We still call them for the 4 weeks to see if anything came up. Maybe they got the report card back and something was wrong on that because they didn't get a test in or something because of the concussion, and then the parent might have to go back to the teacher. But for sharing this with y'all today, I just did if the child had reported symptoms. So 100% of parents, of course, communicate with medical. We're recruiting them from medical appointments. So you would expect that. And then for medical, you kind of see this dip in week 1, but then it increases in week 2. That's just follow-up appointments, usually 2 weeks later. And you see that again to week 2 to week 4. They come back again in 2 weeks. Really haven't found much of parents like calling the medical professionals in between appointments. Maybe it's they know they have an appointment in 2 weeks, so they can ask their question then. They're not really trying to get at questions in between. And then sport. So for sport, I only included children that were athletes. That percentage is out of parents whose child is an athlete and if they communicated or not. So, and it decreases over time. And then school is actually less of a percentage in the initial week. So really initially, not as many parents are going directly to the school and communicating with anybody. Week 1, still decreasing. And so frequency across all weeks. So I added up all the counts of communication, and this was from initial until week 8. So this does include those that were prolonged. And it was an average of about 9 times communicating with the school, 10 times with sports, and then 7.8 times with medical professionals. But kind of big standard deviations here, so there's a lot of variability. And this is communication with school personnel. And so this dark blue line is teachers, so they're really going to the teachers the most. The largest percentage of parents are going to teachers. I find this light gray line interesting. That's school nurse. So ones that have kind of medical training, parents aren't going to as much as those that do not have medical training. And kind of one idea I have for that is it's things like reduced rest breaks during class, or is one accommodation that's often given, or reducing homework, not taking tests. So parents are probably telling the teachers that first instead of the school nurse. But I was, again, just kind of surprised that we're at least notifying the school nurse if my child comes into you with a headache, here's what's going on, that sort of thing. Principal, I was surprised how many go to the principal. And this, again, this is percentage. And this is the secretary. So like I said before, more than we thought were going to like a school secretary, office aide. So the themes and topics of communication. So with the school, is really just informing school personnel about what's going on, the academic accommodations prescribed by the physician, explaining absences. Lots of times with teachers is concerned about missed work, making up the work, how it's going to affect their grades for the quarter, and then just asking personnel to watch for symptoms in their child. Then with the support system, progress updates on recovery, when they can get back to play, if they've been cleared to return to play. And then a lot of parents have said, I kind of talked to them about taking extra, looking out for my child a little bit extra than they would, even if the child is back to play. So just precautions to take while the child is recovering. And then with the medical system, just scheduling appointments, coming to appointments, starting return to play protocols, and then getting those school accommodations. So some barriers to communication. So we are asking them this every week. Were there any barriers getting in touch with who you need to get in touch with? So some parents have said like they're the middleman of communication, even though it's a school team and the athletic trainers at the school some. So why am I the one that's like notifying all the teachers, etc.? Unable to speak with a principal or guidance counselor when they came in to like drop off all the paperwork with accommodations. There is a few parents had issues with principals, just not being understanding. Long response times from school personnel. And then one parent said the guidance counselor said they were going to notify all the teachers, but then some teachers said they weren't notified. So that was listed as a barrier of communication. For the sports team, poor response time from the coach. Athletic trainer should have maybe taken more of the initiative there. The athletic trainer did not reach out to inform or discuss the concussion. Parent informed of the concussion via a note brought home by the child. That one parent was not happy that she just got a piece of paper saying her kid had had a concussion. And then one parent said she, like, if she wanted to get in touch with the coach, she had to like go through a team mom to get the coach's phone number, which is kind of crazy. And then as far as barriers, communicating with medical professionals, misunderstanding that clinic staff were going to be like calling and checking in on them. The parent maybe didn't realize that she needed to kind of initiate that. Waiting on the return to play clearance after the return to play protocol was sent in. So they have to like go through these stages, and the physician has to like sign off that they went through the stages so that they can play. Longer response time than expected, and then just kind of being hard to get in touch with sometimes. And then kind of just overall satisfaction with the systems of care over time. Overall, most parents are pretty satisfied with the school, the sports, and the medical. School had a little bit lower than the other two, but overall, these systems of care are doing a pretty good job. And then this is just showing days to return to learn correlated with some of communication with the school. So you kind of expect them to be correlated with each other. More communication is going to occur the longer the kid has academic accommodations in place. So that's what this is showing. And then, so some conclusions from that data. Most parents appear engaged with multiple systems of care, at least in the first few weeks after injury. Overall, most parent participants are highly satisfied with care across systems. Really not that many barriers were reported. I read those off, but only about 5% of parents reported barriers at any point in time. Communication with school is correlated with days to return to learn. And then just kind of the overall big picture, why are we doing this? Understanding parent communication with systems of care can help us develop better policies for this return to learn process, clinical practice, and efforts to maximize recovery. So some limitations. So we were recruiting from specialty sports medicine and concussion clinics. So most of our participants came from UAB Sports Medicine at Children's, which has like a concussion clinic. We've also had a few participants come from different sports medicine clinics in the area, Andrews and South Lake Orthopedics. So the parents that are taking their kid there might be kind of more hypervigilant parents that really want their kid checked out. So might be ones that are going to communicate more with systems of care. Just different factors related to the particular system may predict communication instead of any parent or child factors. All this is based on self-report, so if they remember who they talked to and how many times. And then also important to note, communication can be a positive or a negative thing. So if things aren't going well at the school, then the parent might be talking more, but if the parent's just like checking in, then so it's hard to tease out what's positive communication because there's a problem, or what's negative communication because there is a problem versus what's positive. But since we are getting the topic of communication, we can go back and like look at why it was they had to communicate. So problems and solutions throughout this year of doing this project, we've been doing it for a little bit longer than that, but as part of being in this TL1. So like I said, we have been recruiting for a few different clinics. We were having trouble getting numbers, getting participants from just the children's clinic, so we reached out to others. We expanded the age range for my project down to, we were just doing 13 to 18, but then we increased it down to 7. But as you saw, I only had like one kid that was in elementary school. So there's missing data from follow-up calls. So kind of on the front end, we're really explaining better to the parents. We're going to be calling you every week. When can we reach you? Is that going to be something you can do? And then just being flexible with participant schedules, calling them when they're free, or just being flexible to keep calling them to get that data. And then there are some differences among the research assistants and recording data. So we kind of problem solved that with our mentor, and we like made some guidelines of how we needed to make sure we're both on the same page and both like kind of collecting things the same way, since a lot of this is qualitative. So then some like lessons learned. Conducting clinical research isn't easy. I know many of y'all know that, especially our problems with just recruiting, getting enough people in within the time we wanted to. And then of course, just following up with them, people hard to get, sometimes weren't able to reach people some weeks. I learned a lot about the IRB process, submitting amendments, and all that good stuff. Nuances of participant payment, learned from this. And it's important feasibility. We might have realized sooner that it would be hard to find participants within this 14 days from the clinic we were recruiting from if we had maybe done a little bit of feasibility pilot testing there. The importance of networking, so reaching out to other clinics, really working with the folks in the clinic since they're in there so much and trying to recruit their participants, networking with the physicians, and then presentation skills. We're doing presentations like this and poster presentations. And I would like to acknowledge all the places we've been recruiting from and the physicians there, the TL1 grant for helping fund this, Dr. Laura Dreer, my mentor, Jillian O'Neill, who spearheaded this project that I kind of built my own piece off of. She's another PhD student, and then my committee members on my thesis. And that is it. Yes.
Great work.
Thank you.
Very important work. So both my parents are school teachers.
Okay.
My dad is a coach. My mom is a teacher. So there's that communication there. But my hometown is so small. Pre-K through 8 was only 600 students with about 80 faculty staff members, which is pretty small. So everybody's phone numbers in the phone book, our phone book constantly rings. So there's that close-knit communication. Mm-hmm. So did you guys look into doing more rural areas, or?
So kind of just like you explained, we have had some people from rural areas where mom's best friends was a school nurse or mom's really good friends with a teacher. So like, yeah, I text them back and forth every day. So like, how do you kind of code that as like counseling communication? So I would say, well, how many like conversations do you think you had? And then I would try to tally that up, but definitely have observed that, or like, they didn't have to inform anybody at the school of the concussion because they were all at the game and saw it happen. So yeah, that's a good point.
And then another thing, I saw that you said a lot of them had a history of ADHD.
I think it was like 25%.
Yeah, so looking at their communication, so frequency of communication, did you kind of look at how often they were communicating before the concussion? So for example, my mom talks to the same parents all the time.
Right.
So was the concussion the factor, or was it just because, they needed accommodations prior because of
So we are asking them, does your child have like an IEP, a 504 plan, or special school accommodations? I didn't look at it for this off the top of my head. I think maybe 1 or 2 have an IEP already. So one that's currently enrolled I know does, and Mom said, I talk to the IEP teacher every day. So yeah, we are getting that, and Mike should be included.
Very interesting.
Thanks. I don't know who to call first.
So you mentioned that you were looking into basically the parents' commitment to being able to communicate with you or the study team. Were parents and children excluded on the basis of not being able to commit to answering questions or the number of study calls, I guess?
Right. I think that once I explained it, they wouldn't enroll if they knew they couldn't do it, or they're like, oh, I can't do phone calls that often. Like, I don't want to be a part of it. We would pitch the study to them and then see if they were interested. Does that answer your question?
Yeah, and to follow up, I'm wondering about some of the implications of not including those folks. Like, might they differ substantially from the folks who are able to make that commitment?
That's a really good point. If they're not interested in doing a research study, then does that mean they're really not the ones communicating with the school either? Because that's a time commitment.
Or are there certain limitations to their being able to?
Yeah, I think that definitely should be another bullet point on the limitations tab for sure. This is also a highly educated group of parents as well. So might not be as representative of overall concussion management.
So I think this was a very nice study. So I have one question and one comment. So the question was, do these schools actually have school nurses?
Most of them do. In the more rural areas, there's like a few school nurses that rotate through schools. I don't think we're asking them, does your school have a full-time school nurse? So maybe that's something we should have been asking all along.
No, I think you have. She's got a mentor on this. He has a zip code, and we can find what area they're at, and we're working with the Department of Ed, so we know what school coverage the nurses are. There are some that are, what we understand, the school nurses cover 3 different counties, so they're kind of strapped. So we'll have that information.
Yeah, we can go back and find that, I think, based on the school information.
Because a lot don't have that, or that as you discussed, there's some rural counties where they are talking more than we thought. They're close there, and it may be a good thing. So it's one of the things kind of just understanding what is going on, how often they're connecting and whatnot. And then sort of presentation comments. I think there's a lot of text on your slides.
Okay.
So thinking about maybe particularly for the if they're really important quotes, I think blowing them up, because I'm trying to read your slides and then listen to what you're saying, and so you can have more slides with.
Okay, for the barriers and topics of communication, so maybe one slide per system instead of all together. Okay.
Yeah.
Thank you.
And then also thinking about contrast. So I mean, it's hard because what you see on your computer is not necessarily what you're.
Yeah, I was when I was looking
The
there.
text on my background was really hard for me to read back here.
Yeah, so one sort of trick is, particularly for your, when you've got multiple colored lines on a graph, is if you print it out in black and white and you can't see it, then it's going to be really hard to parse on a projector. So.
Okay. Yeah, sorry about that, everybody.
Oh, no.
I appreciate that.
I do all the time.
So I just want to say to all the fellows today, just great job in general. I remember my first presentation. I remember all my presentations, and they weren't nearly as composed. And so just in general, I think we always have questions and comments, but overall, great job in general. My first presentation, I don't even think I had slides for the record. I think I was I don't know what I'm doing yet, guys. I'll hopefully figure it out. And so you were just miles and miles away from that. So I thought I would throw that out there too.
Well, this was my wrap up, so I at least had to.
Like, I remember like my last presentation, and again, I don't think I had this like clean cut story. I was like, hey guys, this is what I've done over my few years.
Yes.
So the pictures and graphs and cartoons was extraordinarily helpful.
Okay, thank you.
So if you cut a little bit of text without cutting it too much, but keep those cartoons and so forth to converse. If you're doing a national presentation on a podium for 10 minutes, you don't need those cartoons, perhaps one or two, but so that depends on the purpose. The question, so I understood everything in the perspective of it was crystal clear who your population was, the domains, the research project, I mean, all of that was very clear. I was wondering, at the time you get to write a paper or your manuscript, if you can enhance your design, because it's crystal clear, with some type of conceptual model from the communication lens. So thinking about, yes, we use this conceptual model, and this is how we design our study.
Okay.
It depends on the area that you want to publish or support, but asking or building your research based on a conceptual model, it's critical to say, okay, we did this not because I wanted to do it. Yes, you wanted to do it, but all of that is based in the conceptual model.
Okay.
So I would encourage you to ask your mentors to say, which conceptual model did we use? And then try to embed that.
Okay, thank you. That's good suggestion.
Did you use that conceptual model?
No, I didn't.
But your mentors will have that in their mind to say, this is what we thought.
And I mean, my mentor has done a lot of like qualitative research, like focus group stuff, like really working on like the family perspective. So we tied that in, but I don't think like I could cite a certain conceptual model at this point.
You'll find one. So.
Yeah. Do you have anything to add to that?
Yeah, this is part of like kind of building off some prior work. So there's a general model, but some of it was just understanding where we're at and then what model would best fit, like an intervention, design, or program to help alleviate some of the barriers or better enhance communication. That's a big, unfortunately, big issue, like getting that communication quickly to the schools in between systems of care.
Well, thank you for your attention. Oh, one more question.
Sorry, I just have one last comment. I know that this is the design from the outset of the project was to be very parent-centered, but in regard especially to the like the barriers to communication, I can imagine that some of those barriers could be flipped from the sense of these are barriers that the parents have to communicating. Well, for example, what are the barriers that the coach has to communicating with, the teachers have to communicate with, the school themselves has to communicate with the parents as well. So if you wanted to, I guess, expand the study, is to look at another perspective from what are the challenges that the school has with dealing with this as well. And who from the school, is there a point person at the school that should be communicating this? Is this actually the responsibility of the nurse? What challenges do they have towards communicating with the parents as well? So I think that could be probably equally as important as challenges and ideas from the parents.
They probably don't have time.
Yeah, that's one of the things we've already done. So this is kind of building onto that. This system of care, the parent. And it's hard for the thesis to get everybody, you know.
Sure, yeah.
But yeah, it's a great question.
You know, when you start a study, you set a subset of an idea, what you look at, and then of course the questions.
Yeah, that's good.
Okay, great. Well, thank you for your attention.
Thank you everyone for presenting today, and we'll have more presentations next week. I didn't know that today.
Okay, cool.
Yeah. Oh, yeah. And I don't think I wonder. And I don't have something to review.
No, I just very quick reviewers so my kind of area was pain and opioids, but so when I saw the Prostaglandin, whatever, gosh, I got a drug company on that. Because I remember like when I had to